workshop. So moving on now, we do have the workshop on controlling fermentation and inoculation. And please welcome Ms. Amelia Sage from CQI. Emma, excuse me. Emma Sage, I'm sorry, from CQI. Thank you.
Hi, thank you. Okay. All right, we're going to talk about fermentation. This is something that CQI, the Coffee Quality Institute, teaches as a part of our post-harvest processing program, but is also just a very interesting topic in the coffee world that we live in right now. I have a lot of slides, so it's a good thing I'm the last one. I don't know, you're going to have to hook me to get me off the stage. That's how this works. Okay. Why do we care about fermentation? Well, of course, it's because we all love coffee. And although fermentation has a physical purpose in some instances in producing coffee, it influences the flavor. And that affects the quality of that coffee. As coffee people, we've been trained for generations that we're searching for unique, interesting, special flavors, and fermentation is a big part of where those flavors originate. As I said, before we talk about fermentation, we should talk about processing, because processing is now I've done it. Processing has a very functional purpose, right? It's to take the fruit off the coffee and to dry the seed. We all know this. And to make sure the seed achieves a safe moisture content. But really, there's another reason that we like fermentation, and that's because it creates differentiation in flavor. It can bring attributes to the cup. So although fermentation has happened in coffee since forever, since in Ethiopia and Uganda, the purposeful manipulation of fermentation to create flavors is relatively new. Okay, let's talk about a few science-y things here. I'm a scientist by training, so get ready. I'll try my best. To create flavor during fermentation and during coffee processing, there are a few things that happen. One, microorganisms create compounds which move into the seed, which influences the flavor precursors in that seed, right? It's microbes. Then there's another way that flavor can be created or changed during processing, and that's leaching. Believe it or not, seeds, so coffee, right, can lose compounds via leaching during the coffee processing. And that means, right, there's a lot of water around coffee, especially in the wash process or if it's being soaked in water. And the water can actually pull out compounds from the coffee. That's leaching. The other thing that's going on is that the seed is alive. And therefore, metabolic processes are occurring all the time in that seed. That changes the chemistry of that seed. And so that means that the flavor potential of that seed, that coffee, changes as well. And of course, coffee is complex. So all of these things are happening continually and interacting together through coffee processing. And they're influenced by the environment, right? External factors. It's influenced by temperature, oxygen availability, humidity, pH, and water activity. So within the seed, all these things are going on. And then external to the seed, there are factors that influence them. That's why coffee is so complex and probably why we enjoy it so much. Fermentation happens in all coffee, right? It's not simply a step in the wash process. Fermentation is not a processing method. You can't stop it because microbes are everywhere and are influenced by the environment. We can think of fermentation as microbes converting food into energy, and they leave things behind. Those are byproducts. And therefore, it's a metabolism. It's a microbial metabolism that's happening. So, this microbial metabolism is influenced by the environment, and there's typically a sort of succession that happens during the coffee processing, which is, depending on the environment, especially temperature, oxygen availability, and pH, which all change during coffee processing. And then, of course, we all know that if fermentation goes too far, or if you have the wrong microbes doing the things that you don't want in your coffee, you're going to have problems. Molds or other fungal problems that lead to defects and off flavors, those unpleasant acids like butyric acid, which we really don't want in our coffee. There's a huge diversity of microbes that are present in coffee processing and therefore contributing to the fermentation that happens in coffee. Lots of different bacterias, some of which are really familiar to us at this point, like the lactic acid bacteria. Then there's fungi like yeasts and other kinds of fungi, which are typically the unwanted fungi, the filamentous fungi, which is sort of like the molds and things like that. And really, when manipulating a coffee or when processing a coffee, producers can, change that environment, therefore changing the microbes that are doing different things while coffee is being processed. So it's all about getting the right microbes to do the right thing at the right time. We have bacteria, molds, and yeasts. Molds and yeast are both fungi. Just as a quick aside on yeasts, yeasts are fungi. They are single-celled, and they're capable of fermenting organic animal or vegetable matter. They're small, and they're a very diverse group of organisms. So it's all about controlling the microbes. How do we do that? Microbes can be controlled directly or indirectly. You can indirectly change the environment, right? You can remove oxygen, you can put other substances for the microbes into the coffee. You can alter the pH of the coffee, warm or cool the coffee, or extend or shorten that fermentation time. Those are all external things that can be done in order to manipulate the microbial population. Or directly, one could pre-clean and homogenize the raw material of a coffee. Some producers go to the extent of even sterilizing that coffee. And then there's the opportunity to inoculate, which means you're directly changing the community by putting something else into it. So what does it mean to control fermentation? You have the right microbes winning. The fermentation is producing byproducts that you want, that are good for the coffee, for what the coffee is that you are trying to produce. And we can track microbes and by their activity or by their byproducts. Different microbes prefer different environments, and these are some examples here, but here you'll see a list of different types of microorganisms. There's different fermentation pathways that they use. There's if they like oxygen or not, their ideal pH and temperature. So each one has a perfect environment that they would like to exist in. Notably, very notably, One of the big things that is being manipulated now to control fermentation quite commonly is the availability of oxygen. You have anaerobic or aerobic environments, and that can change the type of microorganism that is part of the fermentation. Typically, each type of microbe goes through this growth curve. When there's a time where that microbe is active, the population starts small, but they start to eat, they start to grow, they start to divide, they eat and eat and eat, and either something about their environment changes or that population eats all the food, right? And then the population declines again. This can be manipulated. Temperature, right? We can use temperature to either slow down or speed up fermentation. Oxygen. This is one of the biggest factors that can change the flavor precursors in a coffee. You see this happening more and more now. Bacteria thrive in aerobic conditions. Yeast thrive in anaerobic conditions. Mold prefers aerobic conditions, so with oxygen. More about an anaerobic environment. This is a term that's used a lot right now in the specialty industry, anaerobic fermentation. And the question is, what do people mean by it? What's the definition? What's really complex is that we know that coffee is not homogeneous, right? Even one vat of coffee, there are differences within that vat of coffee. Even if you do a wet fermentation, which is, covering the coffee with water for the fermentation, it's not a completely anaerobic environment. There are pockets of more or less oxygen available to the microorganisms, and they will find it. In life, right, in nature, there are areas that are higher or lower oxygen availability. Even within water, there is dissolved oxygen. And in most situations, both types of microorganisms, those preferring anaerobic and aerobic, are present. It's just a matter of which one happens to be dominant at the time. So even trying to control to an extreme level the level of oxygen in a fermentation, there's going to be a heterogeneous environment. And all those microbes are still there. Another thing that's within the industry now is this term carbonic maceration. It's a term borrowed from the wine sector, and generally it just means in the coffee sector that there's a whole fruit fermentation happening with low or no oxygen. It's a type of anaerobic fermentation. Now, there are lots of other things that are done in combination with this sometimes, this carbonic maceration. So I always tell people, if a coffee is said to be anaerobically fermented or carbonically macerated, just ask questions. What does that mean? What was the environment like? What was done? Was it underwater? Was the oxygen evacuated, or was it flushed with nitrogen? Was it put in a plastic bag? Was it in a stainless steel tank? There's so many options. And all of these will change the microbial population a little bit differently. All right. I already mentioned this underwater fermentation. That's a very easy way to minimize the amount of oxygen available during a coffee fermentation. You can also try to minimize the head space or the surface area, right, of a fermentation. If you have a small surface area, you're going to have less oxygen available versus if you have a large tank with a big surface area, the oxygen is getting in everywhere there. So ways to minimize the headspace of a coffee fermentation is using plastic bags or just changing the shape of a tank. One can cover and remove oxygen by flushing, right, with another gas, or kind of mix these methods, but all of them would be maximizing the anaerobic environment and therefore encouraging those types of microorganisms to thrive. All right, now what's inoculation? An inoculant technically is an introduced microorganism which stimulates the production of certain metabolites, often for specific purposes of contributing to the quality or the character or the flavor of the product. It's sort of like a starter culture when we think of yogurt or even a sourdough bread or something like this. This is a starter culture. And it can provide particular characteristics in one or more ways to control fermentation. Now, coffee people have been doing this, again, for generations. We know it as backslopping, right? You take a little sample of one fermentation tank that's finished or almost finished, and you use it to sort of jumpstart the next vat of fermentation that's happening. That's backslopping. It's a sort of inoculation. We're adding a different microbial population to a new microbial population. There are all kinds of ways to inoculate coffee now that are popping up with different sort of cocktails of local or native yeasts that have been isolated, analyzed, and combined with the intention of that microorganism dominating the fermentation. Yeast inoculations are the biggest commercial available product at this point to inoculate. This is adding yeast in different ways during the processing, and the purpose is multifaceted. It can be very functional. To inoculate a coffee could just speed up your process. You know, if there's a sort of space issue at a mill and there aren't enough fermentation tanks or something like this, maybe speeding up the fermentation would be really advantageous during harvest. Maybe inoculating would be something to try. It's also something that people try in order to just even consistency of lots, because that microorganism that's inoculated typically dominates the population. That makes the lot more similar, right? More homogeneous. And also, putting an inoculant into a coffee fermentation can serve to protect the coffee from the bad microbes, right? If you are outcompeting the things that are bad, then you have less likely of having problems caused by mold or other fungi that you don't want in there. And then there's also the intrinsic purpose, or the sort of more esoteric vision of controlling the flavor. Yeasts do have very unique byproducts that they impart on coffee, and can be used as a really interesting way to manipulate flavor and attributes in a coffee. All right, so how inoculation generally works is that first the coffee is prepared by pre-cleaning and washing. The inoculant is chosen. Those are specific microorganisms, and then they're administered to the coffee. Now, there are lots of different methods to do that, depending on the processing method, the variety, the species, etc. And it's really important to follow proper procedures for this. And this inoculation will give an advantage to certain populations that will quickly, in theory, outcompete the other microorganisms that are doing their thing at the moment. This inoculant as a commercial product is usually brewer's yeast, Saccharomyces cerevisiae. It grows really rapidly and is well studied. It's used in wine and baking and lots of other industries. It produces the classic fermentation, which is the low oxygen alcohol fermentation, right? It starts with a molecule of glucose, a sugar, and the fermentation will enable the production of ethanol, carbon dioxide, and energy. But At the same time, there are other byproducts that are created, and those end up being flavor precursors. Usually a fermentation will take place until there's no sugars left. I highly doubt that happens in coffee. There's plenty of coffee pulp available. All right. This is a little cartoon showing another thing that yeasts do, which is reproduce, right? Why does any organism eat? It eats to grow. It eats to reproduce. And that's what yeasts do. Some of the sugar that the yeast cells consume, metabolize, is used as energy, but then others are used for reproduction, for growing the population. Yeast are generally have a optimal temperature between 25 and 30 degrees, although they'll die off if it gets really too hot. And they tolerate a wide range of pHs also. All right, so if one goes through the whole process of intentionally inoculating a coffee, you're not done. You have to consider, even if you manipulate the coffee mass, there's the environment around that's going to influence those microorganisms. Also the species and variety. There are different amounts of pulp on different varieties. Certainly Robusta and Arabica react really differently to the fermentation process. There's also whatever the yeast strain is that's being used, or the cocktail of different yeasts. Then there's the methods themselves. There are different methods of adding the yeast. Usually there's some kind of a rehydration of the yeast, followed by an application to the coffee. You can even do this on a natural process. And the protocol, of course. If one decides to use inoculation, either for flavor or for consistency's sake, of course, one has to use the same protocol each time. Otherwise, that consistency or that benefit really is not going to be reliable. All right, so in theory, yeasts create these byproducts that lead to fruity esters, aldehydes, lactones, and ketones. These are all sort of exciting attributes in my opinion, right? The fruity notes, apple, pineapple, citrus. Aldehydes are sort of green and floral. Lactones can be sweet. So in theory, yeasts can impart this type of flavor. But of course, we know that there's a lot of steps in between that fermentation and when someone drinks that cup of coffee. So let's think about potential pros and cons of yeast inoculation, right? Because some things I've said so far seem like pros, but it's not the total story. As I mentioned, it could shorten fermentation time, which can be an advantage during harvest. Also, there's the homogenization that could occur, and defect prevention, and the flavor, right? But on the other side of things, there are some things that to be aware of, right? There's trial and error. It's a new technology. If one isn't used to using it, one has to create a protocol. There's going to be fits and starts. There's going to be mistakes. These things need to be worked out pretty much on every farm that tries it. And that means that the producer needs access to information. Always a barrier, always something to keep in mind. What information is needed? How do they get that? How do we make sure, if we want to try something new in processing, that we're using the most up-to-date information? It costs money, right? It's a new tool. It's a product. That's something that needs to be factored into the business model. So, why? Why would someone innovate with fermentation? Why would someone try inoculation? Well, sometimes it's about rising above the competition, right? There's a lot going on in the market right now, in the specialty market, regarding experimental processing methods, and Those can be big wins. Those can get rewards. Sometimes there's a specific request from a client. That could be a reason to innovate. Sometimes diversifying the products or the coffees available and the flavors available in those coffees could mean you get access to a new market. That's a motivator. Of course, we're all hoping that with extra effort, the premiums will be higher. And then, of course, what I like to call chasing the wild flavor. As coffee people, we love the next new flavor. We want to find unique flavors. We want to taste those new attributes. And those passionate about coffee, sometimes this really motivates them to do something new. Maybe someone wants to be a leader, to push the market forward, to try something new and to be an innovator. The fame and fortune of being a coffee producer. On the other hand, sometimes having good control and good processes is much more beneficial and much more better business call than being the innovator, right? Each producer, each person that decides to take a risk on something new, a new technology, a new process, needs to consider this really carefully, because innovation comes with risks. So to kind of close this up, I really want to emphasize that innovating with processing, changing processing methods, trying a new processing method, using inoculants or something else, who knows, using a co-fermentation, there are all these things that are popping up. All of them should be a solid business choice. There should be a secure market for that coffee and a buyer for that coffee. The risks should be assessed and identified for doing something new. The inputs or the costs should be assessed. Is there new hardware that's necessary? Is there more labor involved? There should be trials, right? When trying something new, try it out on a small scale first. Repeat it. Repeat it again. See what happens. Solid protocols and processes, I cannot emphasize enough, are very, very important to learning something new, trying something new, making a decision about something new. And know what the potential rewards are, right? What's the differential? Who's the buyer for that coffee? All these things are a business choice, and every producer has to make that choice. Okay, here's the workshop-y part of my workshop in the last 2 minutes. Right now, I actually have 3 coffees available for tasting, for a little fun. They're over here by the screen. They're labeled 1, 2, and 3. I'm going to go over and get some, and I'm inviting you to also get some. So anyone who's interested in trying something, I'm going to walk us through it, but you should come try it too. Okay, great. We'll give people a minute to get the coffee. Okay, the test is not to figure out which one is hot. Okay, I see people tasting, so I'm going to start talking again. It's fine. You can still get some coffee. All right, apologies for the temperature, but other than that, I want to tell you that this is all the same coffee, processed slightly differently. This is a Colombian coffee. It's a honey process. Honestly, I'm not really sure about this 48-hour fermentation thing. I don't have all that information. Pay no attention to that. Okay, I'm gonna just take number one. All right, number one is a lot of brightness. I'm tasting a lot of citrus, like lemon. It's cold. It's still pretty